La porta in fondo così, si siamo qui dentro.
Dobbiamo rientrare da qui sì.
Quindi allora risolto, per caricare la settimana passata, avevo caricato nel calendario. Quindi è qui che questo si trova. Però lì non c'è più la scrizione. C'eravate perché c'è la camera, eccetera. Sì, è stato. Dopo lì poi dovevate la Siamo, sì. Buongiorno, iniziamo questa quarta giornata di lezione. Good morning. Good morning, Silvia Franzellitti. Benthic invertebrate as global sentinel of emerging pollutants in the marine environment.
Okay, good morning.
Grazie Silvia.
Grazie. Okay, good morning everyone. I want to apologize about my voice. I'm a little bit sick, and so I take my water here, and we will do some breaths, maybe some more breaths than what we did in the previous talk, because otherwise I think I cannot be able to take the 4 hours of talk. Thank you. Okay, so first of all, just talk about the requirements for your PhD. I decided to, for those that have to perform the exams, that I don't know at the end if will be just the psychology or the rest of the people, I don't know. But to make the thing smoother, for you and also for me, I prepared an exam on Microsoft Forms. I don't know if you know the software, but you have it on the suite of Office that you can reach with your institutional account, the Unibo account. So I can also show you, because I hope yeah. So I will open the exam, I think tomorrow, and it will be closed on May 9, that is your deadline. So you can complete the exam, with reasonable time, I guess. There will be some short open questions to fill, of course, on the topics that we are going to see this morning together, and some short biographical research. So something in the middle between simple quiz and an essay. You know that the exam is just pass or fail. I put as a mark the 50% of the correct questions, but we will see when I will have all your results. Of course, if there is something that you can understand, or if you have questions, you can stop me when you want, when we will talk about. I will send you an email with the instructions, so it will be easy. Okay, any question on this? No? Good. So the second part is to introduce myself. Someone knows me because we have some of my previous students at the marine biology course. Someone that I know for our diving activity together, and Naomi. And my name, as Mauro said, is Silvia Franzellitti. I am an associate professor at the Università of Bologna. My department is the same of Professor Alfredo and Professor Rocchetti, but I work in the campus of Ravenna. So for those that are not aware, how many non-Italian we are here? One. Okay. So you are the ones that maybe are not really aware about how the geography of our region is. So we are here, we are in Marche region, but the University of Bologna is spread all over the region of Emilia-Romagna. So we have the campus of Bologna, that is the biggest one, and then we have several other campuses. For example, in Rimini, is just minutes from here, and in Ravenna. Ravenna has been maybe one of the first campus to be established, together with Zambia Emilia, where is the Department of Veterinary Medicine. And as you can see in the picture, we are in a very beautiful place for the historical and also for the natural heritage. So the main vocation of the campus of Ravenna are the cultural heritage, but also the environmental issue. Also because we have some environmental problems all over the city and also close to the city. Also related to the quality of the coastal environment that has been quite heavily impacted by the previous industrial activity that was established close to the port of Ravenna. So there is a huge industrial area that in the 80s, 90s produced a lot of plastics, polymers, and also we have the hub for the gas extraction production that is in the gas. And this, of course, produced a lot of environmental impacts that we studied. So we are in our research place. We are more or less all involved in the research on environmental risk assessment on land and at the sea, and also on the effect of climate change and other environmental issues. My laboratory, in particular, my research group, that is the Animal and Environmental Physiology Laboratory, works on basically on the effect of climate change, environmental pollutions on animal physiology, and we work basically on invertebrates like mussels, crabs that are enormously abundant in our area, but also we did and we do some research on fish physiology. And we also have some part of the research that is based on the toxicological screening of the pollutants that is related to the human health. So we use human cell lines to test for example, the estrogenic effect of environmental matrices and also chemicals that are still not being addressed as sure endocrine disruptors. And you see here the array of experimental setup that we employed and developed during our history. So from the modeling, the in silico studies, for example, on the interaction of fish receptors and pharmaceuticals. In vivo studies, both on human cell lines and also biomass cells, and studies of embryo and larvae of mussels. Of course, with the acquired studies, some field studies, I will show you something, and also measurement studies for some specific research approaches. I think that all of you are quite aware about how I'm talking about. Okay, so you know which are all this part of the research of the biological research, but we will see something during the talk with case of studies and examples. I try to keep you as much as real case of studies or real applications that I could, okay? Just to give you the possibility, okay, that the physiological research on health within the environmental issues. Okay, so today we'll talk about emerging pollutants and the threats to benthic marine invertebrates in particular. And then I will show you why benthic marine environment could be good models for this kind of research. So the broader definition that I was able to find on the emerging pollutants is this one. That is, by the way, pretty old because it was established in 2008. So the emerging pollutants are defined as compounds that are not currently covered by existing water quality regulations. So they are not regulated by law. Neither for the Italian regulation nor for the European regulation that we are, of course, asked to follow. And but they are thought to be a potential threat to environmental ecosystem and also to human health. Because at the end of the environmental studies, the question is always how much this issue can be also potentially relevant to humans and so becomes a health problem for us. Of course, for human health and safety. Okay, these compounds are not regulated by law, but in 2000, the European Water Framework Directive established a list, a watch list of priority substances that is also updated every couple of years, I guess, more or less. That are under special attention, okay, to be likely considered as a potential risk for the environment, not also for us. So there are among these priority substances, we can find different chemical types of compounds. So industrial chemicals, some pesticides, personal care products, pharmaceuticals, nanomaterials, metals, flavor retardants, and also, in particular from the COVID, also biological results. So we can, of course, in these 4 hours, we cannot cover all the spectra of the emerging pollutants, but sorry, I will talk about my specific field of research, that is the plastic, the issue related to plastic pollution of marine environment, and even more because it's a tale for me of that is lasting by 20 years. We will talk about pharmaceuticals and personal care products as environmental pollution. I know that there are some of you that is working more or less in the fields, right? So it's some of you doing research on plastics or pharmaceuticals. One. The microbiologist, I guess that part. Okay. So the other background that we can have that I have to give you is why benthic marine invertebrates. Okay, first of all, because they are my first, they have always been my research models. But this is not, of course, the real game. Okay? But because if you think about some of the vertebrates that I also put in the picture here, many of them are like in the middle between the pelagic and the bentholic chain. So they can account for the degree of pollution of these food webs. Their life strategies fit quite well for the research questions that we have to address, in particular the feeding modes, the mussels or other bivalve subfeeders, so they can grab everything, almost everything. We will see that they are not so numerous. So they can grab the material that are dispersed into the water column. Other bivalves like the plants may account for the contamination of the entire compartment. The research that is lasting since I think the 40 years clearly show that they have different degree of tolerance or vulnerability to environmental stress. So we can also select the specific animal model based on the degree of stress tolerance that we want in our experimental setup. And some of these animals are sentinel organisms in biomonitoring approach related to the environment assessment of the environmental quality of the environment. So we have a huge amount of data and also of samples spread all over the world for this specific class of organisms. Okay, the definition of sentinel organism, maybe you already heard about it, we can see quite quickly. So they are indicator species that are sensitive to environmental disturbance and may develop measurable biological response that provide early warning signal about the interaction between the animal and the environment, and about the fact that in the environment there is some factor that is acting as stressor for the animal itself. So we cannot know the specific stress that working, but we know that the environmental status in that specific area in which we are collecting sentinel animals, or in which we are transplanting the sentinel animal, is acting as a global stressor for the animal itself. There are, as I just told, some differences between different species. The most popular, the most employed sentinel animal worldwide are the mussels and the crabs, of course, the oysters, by the way. The mussels is probably like the gold standard of the sentinel species, because we know many features about its physiology, about its the specificity of its feeding mode. It has a high sensitivity to the environmental changes, but concomitantly, it has also good survival. You see that mussels can survive in environment that may be also quite heavily impacted by human activity, like for example, ports. The most part of the work inside the port is dredging the sediment, but also eliminating the amount of mussels that naturally can harbor on the boundary and port, and pacifically, peacefully survive with that highly perturbating elements. So high sensitivity with survival, there are a lot of measurable responses that can be addressed on mussels. Many of them have been also included in the Italian legislation and also the European legislation as biomarkers of response to environmental stress. We will see something across the field of studies. And so this is probably one of the most employed. And it is basically followed by plants. Plants have some problems in the balance between survival and sensitivity, because according to the species, we can have low survival and high sensitivity, like Camellia gallina. We know that for Camellia gallina, we also have a lot of problems now here in the Adriatic in harvesting this species, because it has a very high mortality rate as long as the environmental became not quite challenging, like during the summer season. And during the summer seasons in which we have anomalies in the temperature or in other kinds of parameters. While the toughest with Pinarum, which is much more cultured in the north of our region, like in Loro, close to the river, and a good survival, so it's much more stress tolerant, but there's a low sensitivity, and it can deliver very few measurable responses. So we can, if we go to measure the same kind of biomarkers in these two species, we can find very different, really in terms of variation, between a normal situation and an altered situation between these two species. So the selection of plants as sentinel organisms must be done with really a lot of care. And by considering well the environmental issue that we are going to address. The other good features of the mussels is that we can employ basically the whole animal cycle. So from the adult to the larva. The fertilization of the eggs is external, so males and females basically release in the environment oocytes and sperm. And so we can also reproduce this fertilization in the larva and performing experiment directly on the embryo. The embryotoxicity assay is, for example, included also in on the REACH regulation, which is a regulation of the European Union to address the environmental risk assessment of all the gods that are produced inside the Union. and we specifically tested one of the tests that are can be involved for the environmental assessment part. And I will show you how this embryo stages may be very good model to address, for example, the effect of some kinds of pollutant on specific physiological features that are much more difficult to study in the animals, like the biogenesis of the cell. So, questions? Are you alive? Okay, very well. It's the phone just to see the timing. Yeah? Okay, so we can start by talking about plastic pollution. I know that all of us knows how much this is becoming a very worldwide problem. It was a problem also in the past years, but since the COVID and the use of disposable mask and other kind of plastic, single-use plastic used for, of course, the prevention of the pandemic, it has become an even more bigger problem because all the plastic that we produced in this last 5 years, in a way or in another way, it has an high probability to reach the ocean. So these are the data taken from Plastic Europe, that is one of the organs that make statistics on plastic production and also production of plastic waste in the European Union, but also worldwide. And so these data are the last from the last report that has been delivered by Plastic Europe, and they are from 2022, something from 2023, so they are quite well updated. And you see the total plastic production global scale that is about 414 million tons of plastic every year. So it's quite a huge plastic production, and you can see also the percentage divided for the main countries that are plastic producers. And here we see also the number of the recycling system. So if you make a comparison between these two data, you see that globally we are able to recycle about the 88% of what we produce. So it's a very small fraction of the plastic that is produced annually that is undergo the recycling systems, and so can utilize. And these are the classification between the use of this plastic and also the kind of polymer that are employed. So the packaging account for the most part of the plastic that we use. And then building, automotive, and so on. I think that there is one item here that is lost, and that can be actually a relevant part of the plastic that we employ, that are the textile. But basically, I think that everything that I'm wearing in this moment has a component or it is totally made by plastic textile or plastic textile derived from plastic. So the number are quite astonishing in a way. Okay, this is the numbers related to recycling or landfilling or recycling by producing energy. We are here, so we are not so bad. There are countries that work better than us because they have maybe also a longer tradition compared to us in the recycling system, and also maybe a better culture, for the people. But we are, I think we are improving year by year. I also see it in my city that every year they have a step forward in the waste recycling system. And also there are also countries that was worse than us. On the global, we saw the number. Okay, so globally, I remember you that the 8% of what we produce is recycled, and all the rest becomes what? Something is employed for the production of energy, and all the rest is landfilled. And then there is the budget of plastic waste that is not considered in this numbers. So all the plastic that is basically abandoned around. And this is probably the most dangerous part, because since it is not delivered to the treatment system, so not recycled, not landfilled, this part can become a waste with the potential to be spread in the environment. So it is estimated that globally, the 22% of the total plastic that is produced ends up in the mining power. So the 22%, the number that we saw to the slide before, it's a huge amount of plastic. Okay, it's true that the oceans is very, very wide, but we are talking about annual estimates, and so the amount of plastic stuff that is accumulating year by year in the ocean is starts to become a real threat. Also because these are very durable materials with a very low degradation rates. And with another problem or issue that we will talk in a couple of slides related to the sites issue. So the bigger stuff becomes smaller particle size, even smaller. We can reach the micro size and even the nano size. And this increase, unfortunately, the environmental concern of the plastic. So small particles, the small plastic stuff can interact in a way with marine animals and with some marine animals, while the micro plastics, can interact in other kinds of ways. Okay, so the fate of plastics once it is at sea basically depends, at least in the first stage, largely depends on the chemical composition and the physical features of the plastic polymer. So there are some polymers that are less dense than water, so they can float on the surface, and others that are denser than water that sink in the water. Each of the two phenomena provides a different kind of environmental issue, because the floating plastic can be transported by the waves and then by the ocean currents, even at very, very long distances from the point source. But the sinking plastics may reach the bottom, becomes part of the deep sea sediments, or be transported by the deep sea currents, and becomes part of this giant conveyor system of carrying plastic pollution that is spreading the plastic basically all over the world. So these are, how to say, scientific estimates. So you see that by going in the deep, we can have different levels, different concentration. It's not really a concentration, different amounts of plastics depending on the type of polymer. The level is very, very, very small, sorry, but the colors are, the light color is polypropylene, then we have polyethylene, and then the polyethylene characteristic, so it's the plastic. So you see that they can distribute differently according to the polyethylene, that is a quite lighter polymer, remaining almost constrained on the first layer of the sea, while PET, so the polyethylene characterate, sink to the bottom, and so becomes part of the deep sea environment. These are data related to the distribution of this polymer in the open Atlantic oceans. To measure the amount of plastic that is in the environment during the year, it has been developed different kind of protocols and different kind of techniques. And the budget that has been tried to be calculated always gives inconsistency, okay, between the different kind of approach employed. So it seems that there is a part of the global plastic budget that is missing. So now that the modeling system are largely being employed to basically quite different kind of environmental issues. They are started to be used also to try to understand how many plastic is in the ocean. Because to date, we don't have a final conclusion on these problems. So we have different estimates, but there is no consensus of one number or a range of number. So they are much more catastrophic, let's say, estimates, and also much more conservative estimates. And this is one of these, the output of one of these models. I can also show you, because there is a website with obviously the user friendly and output of the model, not the R or Python computation that is behind. And this model took the data from the point source of plastic and and also by considering the oceanographic models, so the models for the distribution of plastics, identified the main hotspot of plastic contamination that are here in the North Pacific Gyre, which is an area of convention of the main storage system here. Here, that is another gyre. In the Indian Ocean and West Pacific, where we know that there is a very huge amount of human population, and there are also some problems related to plastic recycling and delivery, and unfortunately also particularly in Israel and Japan. So this area, and you can see here with the color the amount of plastic predicted to be here, maybe not addressing like a sort of obstacle of plastic contamination. That okay, this let's see. So okay, so these numbers are between 2010, the simulation of the model is between 2010 and 2017. So they are quite recent. But the story of this, of the analysis of the amount of plastic that is around the world ocean started actually far before. Okay, here we are in 2013, but also in 2010. We had the first oceanographic campaign to try to collect the surface plastic and make a graph evaluation of how much plastic is in a selected area. This study in particular try to understand the contamination of plastic in the Arctic Ocean. There are now some research of this kind also for Antarctica, because we know that unfortunately for the delivery of the current system, there are some specific areas both in the Arctic and in the Atlantic Ocean that are polluted by plastic. These areas are quite pristine areas, so there is basically the population no very few nanoparticles. So the contamination by plastic cannot be at this level direct, but it must be indirect. And this paper clearly showed this by the direct data based on the oceanographic survey from the estimates of the population, of the vessel density and the population density. That you see is very, very low, because here is the orange, and here we are on the orange. So we are on the very low value of this scale. While the amount of plastic is quite low, quite high, because we are talking about between 60 and 100, or even worse, between 100 and 500 grams of plastic per kilometer square. Take in mind one problem, the sampling problem, because the sampling has been made with the same kind of sampling equipment that is employed to make sampling of plastic. So it causes a constraint in the size of the plastic items that are finished. And we will see after later that this is maybe a problem, or at least it can improve the bias and the analysis, because the very small fragment basically are not considered in this analysis. So this is an estimate, but probably it is an underestimate about the real amount of plastic that is in this area, but this is a general task. It's not related to this specific paper. Unfortunately, a general problem. Then, always more or less in the same area, plastic fragments or fibers have been found also in ice cores. In a layer of the ice cores that have been formed also several years before the sun. You know that the ice cores are sort of memory recording about the climates, but also about the modification of the atmosphere and also of the sea at the time in which the ice has been formed. So finding fibers and also in quite huge amount in this internal area of the core is quite impressive. I think that they went back about at least 20 or 30 years. So coming to the deep sea, we saw that the decimal part of the plastic polymers is to sink, and so be part of the deep sea environment, but we are talking about the very deep sea, okay? Again, there are modeling about the amount of plastics where it has been possible to take the direct sample, so by rove or by rolling oceanographic campaign, and then by loading the real data. And of course, the data are quite impressive because we are talking about kilograms for kilometer squares. Again, the data analysis is the part that has the darker colors. Probably also because it is not so deep compared to the course of plastics. So maybe we have much more accumulation in the environment also because of the oceanographic features, the oceanographic features that we can. And then we have also a dark color here in this part of the balance, where there is a lot of stop. The deepest location in which microplastics have been found is the Mariana Trench. So 10,900 liters. In this case, we have a raw sampling, but not of seawater samples, but of animals. So we start seeing the use of animals, in this case amphipods, as sentient organisms, because the biopopulation of compounds is one of that biomarkers that is can be assessed on sentinels animals. Sometimes the level of I'm talking of water pollutants, plastic are quite different kind of pollutants compared to the one that we conventionally study. But sometimes the concentration of this pollutant in the water can be really low. So also quite analytically hard to be analyzed. But one of the features of the sentinel organism is to have likely a good biopopulation capacity for the pollutants, for example, mussels, powerful biologists, they filter a lot of water, and they have metabolic features that basically don't allow the fast degradation, in particular organic pollutants. So the pollutants are accumulated in the body of the animal and can be analyzed. For the plastic and the fibers and all the plastic items, the story is more or less the same, but the rate of accumulation likely depends on the type of the plastic atom, in particular on the shape. So the textile fibers, for example, are much more difficult to be eliminated compared to the plastic fragments, and also on the complexity of the digestive system of the animal. So the more the system is complex, the more the plastic stuff can remain longer, can remain in the body of the animal and the I don't know who starts on how locate the this picture. After the digestive system of the starcher is for sure much more complex compared to the one of fibers. So probably the capacity of these amphipods to retain the fibers, plastic, is also very peculiar plastic fibers. But in this case, this kind of physiological feature is useful to try to understand if also this very big environment we can find traces of plastic contamination, and the answer to this question is unfortunately yes. Then the Mariana Trench, this sample stand out as the very most pollutant compared to the others, probably because of the oceanographic features of this area compared to other sampling sites, but also in the other sampling sites. So in other trench, we found detectable amounts of fibers. Interestingly, the main fraction of the fibers that has been found in the amphipod are blue, so blue coloration, because in this at this depth the visual system of the animals are specifically adapted to see the blue light. You know that if we go down in the water also by few meters, we lost the red immediately. and then we lost the other colors. And this depth, the light is really blue. It is named crepuscular light. So we have only some wavelengths of the blue field of the blue solids and spectra of light. And so the animal like this depth are adapted to see this specific blue wavelengths and also the wavelengths related to the biodimension. So basically blue fragment. So coming to the Mediterranean Sea, do you want to make a break before the Mediterranean Sea? Yes. Okay, so also at home we do 5-10 minutes of grammar and we become back. Okay, welcome back. Also, Torma, I hope. So we were here talking about the exploration of the Mediterranean Sea. You can find in the literature, I think, maybe more than 100 papers related to the evaluation of plastic content of the Mediterranean Sea, and I'm talking just about the water, not the organism. But again, by modeling the amount of plastic that reach the ocean, the sea, through the rivers, because in particular here in Italy, we have this problem, and in particular related to the Adriatic Sea. The Adriatic Sea is very narrow and not so deep, but we have one main problem. The Po River and all the other rivers that flow in the Italian border. We now know that just in the Emilia-Romagna we have about 30 rivers, which are all the ones that flowed 2 years ago, and maybe the rest that we saw. But the Po River and the other rivers are really relevant, okay, in making the chemicals composition and also the contaminant burden of the coastal environment of the Adriatic. So by taking this consideration, in this paper, they took as input sources the rivers and also the cities, the bigger cities in the Mediterranean, and they make a modeling of dispersion of the plastic from these input sources to the whole Mediterranean that I'm going to show you. Of course, the main sources are the rivers, we saw the port and other kinds of river, and the cities, Barcelona and Alexandria in Egypt, and here is near Turkey. So these are considered as the main sources. But keep in mind that during the summer season, the population on the coast of the Mediterranean basically double, or in some area becomes 3 or 4 times the number that we have during the winter season. So all this accumulation of people, along the coast, of course, as leaves, okay, it's free. So this is the simulation that the Centro Euro-Mediterraneo sui cambiamenti climatici, it is in Italian, sorry, so the CMCC did. And you see that we can see very well the point sources, in particular here in the Adriatics, and how the plastic should circulate, at least in the surface water. So also here in Adriatic, as in the Atlantic and in the Pacific Ocean, we have the giant. So area in which the current system likely accumulate plastic and not accumulate something else, like plankton and all the animals that float and that have a very low capacity of active movement. And this is, I think, the first element, okay, that is important to understand the biological problem of wastes and also the entry of plastics into the marine floods. I told you that the story of the evaluation of the plastics pollution in the Mediterranean Sea is pretty old. Okay, so it's about 15 years that the Mediterranean is monitored and also quite capillary monitored for the presence of plastic. The amount and also the composition and the size of the particles, we are always talking about surface environment, is not so different from the open ocean. This is a quite very popular paper from oceanographer of the University of Cadiz. They did, I think, 5 or 6 oceanographic campaigns of this kind only in the Mediterranean Sea to collect really a huge amount of data. And now also developed an algorithm to monitor the presence of plastic using satellite data, which is very, it seems the new frontiers plastic monitoring across the globe, and may be very effective also to understand the short-term dispersion of this new kind of evolution. This is interesting because this was a thesis of the master degree in marine biology, which I teach, from one of my students, I think 5 or 6 years ago. That thanks to the Erasmus funding was could go at the University of Cadiz and work with a colleague of the previous monitoring campaign, and she did the evaluation of the occurrence of plastic and plankton in the samples that have been taken here in this area. No, I'm sorry, in all the samples that you see here, highlighted with the extra colored dots. And in particular, the samples that have been collected here have a ratio plastic to plankton that is bigger than 1. So it means that in this area, the animals that hit on the surface plankton have a bigger possibility to hit plastic instead of one. And also the global estimates that we can see in the paper said that in the future we could reach the situation in which in the sea we could have much more plastic than fish by biomass, so by weight, which is really not a good situation to a good forecast. But here, these are real data. And so seeing the real data, it's quite impressive. Okay, with very simple analysis. So counting plastic and counting plankton found in the very same sample of. Okay, so we already see some physical features of the plastic that affect the distribution. But the physical and the chemical features may affect also the way in which these items may interact with the biological systems, so animals in particular. And also the type of polymer and the type, the chemical composition of the plastic may also affect biological effects. Okay, so under a practical point of view, the plastic are the plastic item, like this one, is made actually of two types okay, of chemical compounds. So we have the plastic polymer that are very large molecules that are formed by the joining of monomers. This is quite easy to understand. And it is the main component of plastic. So, for example, we have polystyrene and polyethylene terephthalate that are made by one single type of polymer. But there are also other kind of polymers that are made by a mixture of different polymers, like the epoxy resin that is made of this bisphenol A, which is, by the way, an endocrine disruptor, and epicloidine and variable amounts of other kind of. So, of course, in this case, we can have a mixture of possible components that may have also different biological outcomes. Then, all the plastic are, once they are formed, the polymer are completed with additive. So chemicals that are added to the polymer to give to the plastic specific features, like the durability, like the resistance to microbial attacks, the resistance to UV or to the heat, the flexibility and other kind of chemical or physical features. So actually, a piece of plastic is like a cocktail, okay, of potential of compounds that can potentially interact with the animals. And all these compounds may have its specific biology. So it's speaking as in toxicology, by making an experiment with a piece of plastic and an animal, we are exposing the animal to a mixture of compounds that may have interactive effect by themselves, between themselves. So it's quite complicated, okay, as a Università by a biological point of view. Once the plastic item is released in the water, it can undergo to different kind of transformation that altogether made the weathering of plastic. So weathering in Italian is not possible to be translated. So we are talking in English, so it's fine. I have no problem, but when I have to explain this issue to my students during my normal lessons, so in Italian is always quite difficult to say what specifically weathering is. It is an array, okay, of physical and chemical interaction with seawater that change, okay, the physical stability of the plastic, but also the chemical density. We have, of course, the physical abrasion, the fermentation, the photodegradation, so the interaction with the UV part of the light spectrum, the thermal degradation, the oxidation that is particularly strong in the condition of seawater, because seawater is huge oxidant, hydrolysis, but also the biodegradation, so the interaction between the plastic fragment and the microbial components of water that may change, for example, the physical, the buoyancy of the plastic. So plastic polymers that normally used to float on the surface, if they are covered by the microbial film, may become denser than seawater and that cannot sink. So instead of interacting with the surface animals, can interact with the deep water animals. The problem is more or less the same. And also the fueling of the plastic items with the microbial component that form a sort of film that is called eco-biocorona may change, okay, the chemical perception of the plastic animals towards the plastic items, towards the animals. So virgin plastic, that are the plastic that we take and put in the water, may be not so chemically perceivable by the animals, but if the plastic item is covered by this film, so by this eco-biocorona, it can become like the junk food, okay, for mussels, or also, for example, for seabirds or sea turtles, and so be feeded. So all this process may have, okay, in fact, may change the bioavailability of the plastic material, and also change the size. So the physical degradation, the fragmentation may bring the plastic bottle to become a huge amount of microplastic. So plastic of very, very small size. And the other, say, negative side, okay, the world of the weathering, is that the plastic may leak the additives or even the monomers that form the polymers to the seawater. So these chemicals may become soluble in seawater, and so may become bioavailable for the marine animals. So the leaking and occurrence of plastic additive is probably even more relevant, okay, environmental issue, the more relevant environmental issue related to the pollution of plastic. Okay, so the sites of the plastic litter makes it's biological concern, because different sites of plastic litter may interact with a different array of marine animals. So when we go down the sites, the amount of animals that may be affected by plastic pollution increase. So macro, meso, and in part also microplastics, so going from up to the micrometers scales are sometimes taken up by the big marine mammals or the other vertebrates like fish, turtles, and even okay, animals of smaller size. We are still in this part of the effect of marine litter on marine animals, so on the science that is called the visible effect. The one that probably we saw a lot of time in the documentaries at the TV or also in the newspaper. So the induction of plastic material, the entrapment, the determination of habitat, and also the problem of finding species. In particular, of encrusting marine organisms that can be transported along with, and also microbial species that can be transported along with the plastic item, or also far from its original habitat. So have the possibility to find suitable habitats also in places where that specific animal is not, and become a further environmental issue. The micro and the nanoplastics are probably the more threatening part of the biological impact related to it. Because this small fragment may interact also with very small animals, and also the bigger size animals may easily enter the body, and also easily interact with the cellular components. Nanoplastics, unfortunately, have also the potential to be integrated in the cell, and so interact with the intracellular component like cellular proteins, but even with the DNA, so lead to genotoxic effects. This is why nanoplastics have received a special attention. There are some papers we were talking about with the one of you. There are also some papers that detected nanoplastic in human placenta, and recently also in the human brain. So, and this plastic has been, delivered to our body through the food, with the alimentation. So they are real value. So the research is not is trying to understand, first of all, the root of accumulation of plastic from the environment, and then also the real extent of the environmental issue. So for the moment, we will talk about the microplastics. So we are not going to go so deep in the problem. Okay, another problem that I already told you about is the methodologies that are employed to measure plastic footprint. In particular, when we go too so down in the size and particles becomes not visible, there is a strong analytical problem under this point of view. And the problem here, it is close. There are not three, but there are four main problems related to the isolation and identification that are related to the variability in the environmental matrices, extracting microplastic from seawater, and extracting microplastic from biological tissue. There are two completely different words, and they need two completely different protocols. In the tissue, also the kind of tissue gives some challenges, because in some type of tissue we can perform some types of protocols and others no. But because of really the composition of the tissue, for example, the fat tissues, so the tissues that contain a lot of liquids are much more challenging than what is contained, because the liquids mainly interact with the quantification and identification of microplastic when we use some particular technique like SPR, which is the microscopes with integrated spectrometers, or other kind of chemical analysis or thermal analysis, so like graphic techniques, for example, in which the lipids may the spectra of the lipids or other biological macromolecules may overlap with that of the polymers or the additives that compose the plastics. The microscopic technique, of course, have a size constraint, so we can go until one type of size, but not under. And they do not account, of course, for the chemical composition of the polymer. So we can say that that is a piece of plastic, but we cannot assess which kind of plastic is that kind of plastic. When we talk of environmental samples, of course, the weathering may change the chemical and the physical for sure, but also the chemical structure of the polymer. And so the quantification, in particular the analytical quantification, may become sometimes they cannot be performed anymore. So all these problems are subject of the research of the field of environmental chemistry, because of course the goals would be reaching some standardized protocol to be performed everywhere, and so having data that can be compared to each other. At the moment, if you try to make a comparison with the amount of microplastic collected in different areas of the world, or even with different papers from different researchers, you may have very wide different also numbers, okay? Because of how to say experimental constraints, and of course this is a big, maybe the biggest experimental problem at the moment. Okay, this paper that are probably that I also know. They made a very quick comparison between, for example, two microscope techniques. So the FDR, that is probably the most employed approach for the identification and also the quality evaluation in terms of polymer type of plastic. The Raman, and then a different type of analysis that is the TGA, so the thermodynamic analysis. Here you see that we have more size issues that is present in both in the FTR and also in the Raman. I'm not sure about this 20 micrometers. It seems to be good. I think that the FTR can go also at smaller size, but I'm not sure because I just used it a couple of times. But the main problem for all this kind of analysis is the pre-treatment of the samples. So the real isolation, okay, of microplastic. All these techniques need a pre-isolation of plastic, which with the seawater is quite feasible. With the biological samples or with the sediments, maybe not so simple to perform. Okay, so we cannot have a comprehensive analysis, but we may attempt to make a comparison about the contamination of bioplastic of marine environments. And in this case, we can use, as I said, the sentinel organism and Benthic invertebrates as sentinel organisms. And this is basically the experimental setup employed in this paper that is also quite recent because it is from 2023, in which they collect, make a very comprehensive bibliographic research on world of science, which is one of the biggest contributors for peer-reviewed scientific papers, by integrating, of course, the search by keywords and the eligibility criteria that are listed here. They select 55 studies representing 244 locations worldwide and 412 observation. Okay, so sampling points. From this analysis, and I will show you now the result, they found that at least 141 species of Benthic invertebrates are reporting contamination by microplastics. The most represented are, of course, Mollusca and probably biofilms. And then we have also other species like Stachyra, Invertebrata, Cnidarium. So not only the conventional sentinel organisms, but maybe also animals that are more ecologically, environmentally relevant. Okay, so these are the data. So this is like the superimposition between the data point in which we have the positive sample to plastic occurrence and the worldwide distribution of floating microplastic obtained by the models that we saw already before. With the usual unit measure. So number of microplastic per kilometer square. Oh, sorry. You see that there is a bias in the publication because a lot of studies are focused on the Mediterranean Sea, on the north of Europe, and in this area of. So this have to be, of course, considered. So it is not really a coverage of the worldwide situation, but it is all representing all the data that have been published up to 2023. And if you are curious to understand which are the main bioaccumulators of plastic worldwide, we have, of course, the Mollusca, but with more or less the same average, if we make this data, we have the Cnidariums and also Cnidarium that works more or less like Mollusca. In the paper, they also made a comparison between different feeding modes. I didn't put the result here because the picture is not really, I would say, eye-catching, so it is not very easy to understand, so it's not relevant here, but the omnivorous species are the ones that bioaccumulates more, not the filter feeders. That is a quite interesting result. We are talking about benthic animals, so among them we have animals that feed on the water column and also animals that feed on the sediment. Probably the sediment compartment should be considered much more in the analysis. because probably much of the microplastic that we are looking for, because as I told you, there is a gap, okay, in the world budget of microplastic. Could be in the sea bottom sediments, and animal feed on the sediment will be much more exposed to this kind of pollution. But we work with the mussels, and so we did some research of microplastic, bioaccumulation by mussels. We employed different techniques, like, of course, the microscopic technique, and also the pyrolysis linked to plastic chromatography that is able to analyze the concentration of polymers. In this case, these are all laboratory experiments performed by giving to the adult mussels microplastics made of polystyrene together with the food. The particles were of 3 micrometers or 55 micrometers, which is more or less the size range, the lower limit and the upper limit of the plankton that is fed by mussels. So we gave them something that they could eat. The particles were also labeled with a fluorescent dye that is inside the plastic, not on the surface. So we could follow quite easily the accumulation of microplastic inside the tissue. So here we have the endlymph, so the blood of the mussels, and we saw quite pretty well the microplastic, more 3 micrometers microplastic than 45 micrometers microplastic. So it means that in the recognition of plastic probably sites by mussels, probably sites matters. And here is the digestive glands of the sample of the mussels. So these are tissue sections in which we can see very well, again, the 3 micrometers. The digestive glands is more or less the liver and the and the pancreas of mussels. And here we have the concentration of polystyrene that have been assessed in the digestive gland and in the gills. Sometimes the particles may remain attached to the surface of the gill tissue because the gill tissue has a lot of mucus. But the main accumulation is, and it was expected anyway, in the digestive gland because it is the site, the organs in which likely the vast majority of pollutants are accumulated. And by looking at the data that are in the literature, we can really draw like a route of accumulation of microplastic that goes from the external of the animals to the digestive glands. So the digestive gland is probably the final repository of microplastic and for sure of other organic pollutants. And when they are in the digestive gland, they can be eliminated because we did some experimental trials to see the clearance rates. But the rates mostly depends on the metabolic activity of the animal. So if the animal filter feed, so they are feed with food, they have a normal metabolic activity, and so they quite slowly, eliminate the plastic. If they are kept without the food, they filter feeder with the slower rates, and so also all the metabolism or the digestive activity is slowed down. Other tissue like the mantle may undergo some remodeling across the life cycle of the animal. In particular, during the reproduction, the mantle tissue is clearly dislocated because the mantle contains glands. So when there is the spawning season, the animals release the gametes in the water, but also with the gametes they release most part of the mantle tissue. So then the resting state, they have to rebuild the mantle. So in this remodeling of the tissue, maybe most part of the pollutant and microplastic that can be accumulated because the mantle is a storage tissue may be lost in the water. So there are some constraints by for the accumulation in the tissue distribution of microplastics that have to be considered. Okay, the other big issue is related to the larval stages of the bivalves that lives as part of the plankton and the very first part of their life. And so they can be found in areas in which there is a convergence of the currents, and so convergence between plankton and plastic, like the famous North Pacific gyres that we just saw a few slides ago. So the plankton can be polluted by plastic because in many cases larval stages are active figures. From one stage of their development, they become active feeders. So they can feed on plankton and becomes the first stage of the food webs that can reach also the predators like turtles and other fish, as we saw. For example, in the Adriatic, with Professor Candela, we did an evaluation of the plastic pollution and the potential effect of the internal microbiota of the sea turtles, that lives here in the Atlantic. And the Adriatic is a feeding area for these turtles. And the main feeding object are mussels. So they feed, and also small fishes. So they feed in a food webs that is probably quite polluted by plants, with some also interesting effect on the internal microbiome that probably puts them down next to this afternoon. So I was the focus. Just to say that the larval stages may start a series of events that they have very, very wide effect on the marine fauna. So you see here, these are really nice pictures of our dead mussel that we are able to produce in the lab to the external fertilization of the eggs. And these mussels are 24 hours old. So within 48 hours. So within 48 hours, they can build their first shell and they start feeding. This is why the experiment that comprise the feeding behavior may be established at these developmental stages. So these are the values. And at this stage, the manager it really well, unfortunately, microplastic. You see that we have an increasing amount of internal microplastic treated by the manager that is correlated to the amount of microplastic that we have in these are the larvae. So the post-fertilization the way in which we address the age, okay, of the larva. So we are 48 hours, 3 days, 6 days, and 9 days of specific. And we see that there is an increase of accumulation of unjustified plastic that is correlated with the concentration of microplastic in the water. The microplastics are the 3 micrometer polystyrene that we already see in the apple. The 3 micrometers is a good size also for the larvae. Why we have to use this stage? This stage of larvae is quite important under different physiological point of view, I have to say, because here we have the establishment of the first shells, we have the development of the complete digestive organs, and the onset of the activity. And with the activation of a series of physiological mechanisms like the stress response, also the possibility to degrade the chemical compounds by the action of lysosomes that are internal organisms. And here you see the physiological aspect. At this stage, the only parameters that we can analyze is only biomarkers. So we can analyze are the molecular biomarkers based on the transcriptional analysis of genes that are relevant, okay, to this physiological process that I just showed you. And we can see that the ingestion of microplastic is physiologically challenging for these animals because they increase the genes that are related to the immune response that at this stage are very tightly related to the feeding activity of the animals. And there are some movement of the genes that are involved in the neuro and we'll talk about some of them in the pharmaceuticals. And the overexpression of genes related that are involved in the shell biogenesis, like the synthase and probably because this is the response to the larvae for mitigating possible effect on the whole developmental machinery that comprises, of course, also the cell machine handling. Speaking about the polymer, you see that also styrene alone, not the polymer, but the monomer, may have a quite huge effect. This is the ecotoxicological test that I told you that is included in the REACH regulation. It is quite easy. Yeah? We fertilize the eggs and we expose the eggs just fertilizing to the chemical that we have to test at the increased concentration and wait for the 48 hours to see how many fertilized eggs becomes valid and how these valid are. So if they are normally developed with this D shape, or that if they present malformation, or even worse, if they at 48 hours we have no valid development. So if the larvae did not complete their development. Here we can see that at increasing styrene concentration, the percentage of larvae that undergo a normal development, so if the larvae that reach the D valid stage decrease significantly, starting from the 0.1 microgram concentration, so pretty low concentration. That is also environmentally relevant because in some places of the earth, this concentration was measured in the seawater. And also the larvae were smaller in size. So the development was probably slowed down with some manipulation, but also with a good fraction of larvae that were pre-developed also actually out. We also, of course, did the analysis with the molecular biomarkers, and we saw the physiological processes that were mainly impaired by the accumulation of styrene in the larvae. And also, with someone, we were talking about the BPA, which is an additive of the plastic, but also a component of some kind of plastic, is also the polymer, like the polycarbonate. Styrene has been banned by the European Union, but I think also by in the United States, from the use in the plastic that is used to make toys or other kind of stuff for the baby, because it is an endocrine disruptor. It is really officially classified as an endocrine disruptor. Indeed, if you see in a lot of plastic items, you can find the label BPA free. Also the plastic employed for food material. And indeed, also with our larvae, we saw that the BPA can have effects that are consistent with the fact that it is an endocrine disruptor. Again, we have also the molecular part, and let's skip down because we are in all these effects of BPA are probably related to the fact that it is able to interact with one neuromodulation system that is really relevant to the physiology of muscles, in particular in the early stages, but also as an adult, because it is that is the serotonin system, and the system that relates the effect of serotonin. Serotonin in the adult, for example, control the reproduction, control some part of the metabolism, and it is also believed to have a strong control effect also in the immune. So the fact that BPA is able to, for example, to reduce the number of neurons in the larvae that are able to secrete serotonin gives the idea of the say negative environmental impact of these compounds. Okay, so do you want to make a break, or I can go on? Maybe we can finish it earlier. Are you still alive? See? Okay, so now we skip to the other topic of the talk. The time pharmaceuticals in the environment. Okay, the beginning of the story of the evaluation of pharmaceuticals and pollutants actually started not from the invertebrates, not from the assessment of the environmental levels, but from the assessment of some strange effects and the amount of pharmaceuticals that can be found in the fish. And again, it started from the United States and also Canada. I did the part of my PhD in Canada some years ago, some years ago, and already 20 years ago, at least, they were working very actively on this environmental topic because they had some problems with the fish population, in particular in aquaculture systems. So fish, male fish with female organs or female fish that were not able to reproduce, this kind of physiological effect. And then they started to analyze the amount of the possibility, okay, to detect pharmaceuticals in the fish bodies, and surprisingly, they found a lot of chemicals that are applied as pharmaceutical in the fish, like norfluoxetine. This is the metabolite of fluoxetine. You don't know what fluoxetine is, but if I told you Prozac, maybe you are much more aware. So fluoxetine is the active principle of Prozac. And others. So this has been probably the first, this picture is really famous, you can find it in a lot of websites. So this has been the first proof, okay, that the pharmaceuticals can be found in aquatic natural environment. Here we are talking about freshwater fish, by the way. Meanwhile, the marine invertebrates has always been a source of pharmaceutical rather than the subject of the pharmaceutical. And indeed, we are still using some pharmaceuticals that have been derived by bioactive compound obtained from marine invertebrates like amphibians or gastropods or even shrimp or cnidaria. Many of these compounds are anti-cancer drugs because they have strong cytotoxic activity, so they are toxic that are used as a defense system by the animals and that we are employing as anti-cancer. For example, the first anti-cancer drugs used to treat some types of leukemia derived from, as they derived from marine animals, the trajectory. I don't remember the patent name, but it's one. And it has been identified in marine sponge. It is just after a lot of time that we started to consider the issue of pharmaceutical as a real issue. And only in 2018, we have the first official document of the European Union that recognizes pharmaceutical as an issue and give a series of recommendations to the states to implement the monitoring systems and also to implement experimental protocols for the environmental risk assessment. So, of course, pharmaceuticals are emerging pollutants because their employment cannot be constrained by environmental issues. In some time, many of these compounds save life, so they cannot be restricted for the use because of environment. But we can probably implement much more better, employ use of this compound. And I'm talking about the antibiotics. And we will see all of this. Okay, so pharmaceuticals as environmental, as chemicals spread in the environment have a double nature, because they are synthetic chemical compounds that are released in the environment, like many pollutants, but they are also bioactive compounds that have, in many cases, a specific mechanism of action or mode of action that target, in many cases, specific molecular target inside the cells or even worse, over the cell membrane. So if this target is conserved also in the marine animals that we are considered, the pharmaceutical may have in this animal the same therapeutic effect that it exerts the human patient. Okay? But this is certainly one of course. We want not to treat masters with ibuprofen, no? So when analyzing the physiological effect and the environmental impact of this pharmaceutical, we have to consider this. And the molecular, the biomarkers that we are look at to discover the biological impact of this compound must consider either the fact that these are chemicals, okay, there are pollutants, and so we can employ the conventional biomarkers that we use in the monitoring system to address the structural scores of the animals, but also the fact that these are therapeutic agents and they have a specific function. So, mode of action and specific endpoints. How is perceived the issue in the research field? It is the issue of marine environment contamination by pharmaceutical is an environmental issue since under the research point of view, I'll say, since a lot of years. From the COVID, again, this issue has become even more relevant, and also for the public concern. First of all, because I see that we experienced the problem of the search for antibiotics effective to treat some kind of human illness, and secondly, because I think we are becoming aware about the fact that one of the side effect of the COVID was to spread even more amount of pharmaceutical in the environment. Of course, the release of pharmaceutical from our body is something that is not possible to explain, no? The treatment plant for the wastewater are not made to mitigate the concentration of these compounds because they are not regulated by law. Now there are some new treatment plants that try to also address this problem, but they are still on the tower. And so it is a kind of pollution that we cannot really constrain. If you look in the Scopus database, I looked in the Scopus database last year, and I found about 1,549 papers related to the issue of marine of the effect of pharmaceuticals on marine animals. And by looking at just of the last 10 years, we are 1,062. Huge amount. So we started to try to employ all this data that we have available, to try to see how is the global situation of pharmaceutical population in the marine environment worldwide by using the invertebrate as sentinel. So we use the sentinel organism by making actually a huge bibliographical research. So from all these papers, we built basically two different data. One with the tissue concentration of pharmaceutical. So the sample animals collected in the field assessing the amount of pharmaceutical in the tissue, and a very huge amount of paper on the biological response to pharmaceuticals. So these are laboratory experiments on the effect of pharmaceutical. In vivo, in vitro, on larvae, there is a mixture of environmental stuff. So starting from the field level data set, we set some criteria to explain the research. The data should be referenced, the methods should be clear and control, and this is probably the most important, the data should be expressed on the tissue dry weight, because it is needed to make a comparison with the concentration from different papers and different methodologies. Also, by taking this eligibility criteria, we built a data set with 750 people. And in this case, I really appreciate the power of calculation and integration of our packages. So if someone who is still, like to say, not happy to use R, I suggest you to start learning. I know that it is not easy at the beginning to make all that script, but then you will be happy, okay, to learn. In particular, when you will have to write the thesis or before to write the paper. So by making this integration, we found that the most part of the work is done on bivalves, and we were quite sure about this. But we have also other models that are also quite interesting, like gastropods, crustaceans, and again the diets that stand out to have of the papers, the works in mountains. And as the therapeutic classes, before looking at the data, I thought that antibiotics should be the more addressed in the field, but actually it is not. But the psychiatric drugs, like the product that we saw before, are the more found in environmental samples, and then antibiotic and non-steroidal anti-inflammatory drugs like ibuprofen. So in the last couple of weeks I used high quantity, and then liquid regulators and so on. And this is the distribution of the data. That is still to be refined, because some records are still missing from this mapping. But we can see that, first of all, we have a representation of also high-altitude areas. These are samples taken close to the south islands. And the Mediterranean is always the most represented. So if you are curious to know where the highest concentration was found, highest concentration on average is 700, 12 nanogram per gram of dry weight, tested in coral, aquafora, in the Red Sea. And the compound is sulfomethoxazole, that is an antibiotic, and employed to treat respiratory illness. Maybe you took a lot of that when you were kids, because it's the active principle of vacuum. I heard that. Yes, I also took a lot of vacuum when I was a kid. So it's given kids that like short and so on. And of course, the Mediterranean, you see, it's really a hot spot of investigation in this field. We have 11 out of 24 the 25 studies considered that reported after pregnancy. 11 out of the 16 therapeutic classes that was detected worldwide are detected in pregnancy, and 5 out of the 7 families of animals that have been employed in the studies have been also sensitive to pregnancy. Probably because of the diversity of habitats that require also the possibility to analyze different organisms. Oh, these are the data classified by therapeutic class, but we saw again that the antibiotics are showing the highest concentration. Okay, so the Mediterranean Sea, we saw that is a sort of it is an hotspot for pollination to different kinds of pollutants. And the pharmaceutical is not an exception. And the Adriatic, we know that has a very peculiar oceanographic features, starting from the fact that we have the rivers that affect very heavily the distribution of pollutants, but also the different parameters of the water column, like salinity and chlorophyll. These are the registration of salinity after the last year from the station of Porto Garibaldi, and you see the changes up and down of salinity that are concomitant with the heavy rain of the north of Italy, like in Silvia and so on, and of course with the floods of the Università. So, of course, when we have the floods of these rivers that drain areas that are rich of human population and also of industrial activity, and also of biomedical activity, industrial activity, we could have a rich parts. We did monitoring analysis by employing the mussels that are found on the coast of the Adriatic, so from Bologna to Senigallia, and also other colleagues of the Università delle Marche did the same stuff southern, the same type of analysis southern, and actually we were able to detect different pharmaceutical that are included in this for therapeutic class in the mussels, but also at quite high concentration, because we are about at 160 nanogram per gram. These are the paper. The same similar data are available in the literature of sediment of the same area. But the interesting result is much more on the seasonal trends of these compounds. These are from our monitoring, and this compound, I think that is science, anatomy, probably didn't not tell you nothing, but if I show you what this compound is, we can understand why we have this seasonal trends. So the increased employment of alkane, DHT, is the active ingredient of alkane. The increased employment of the alkane, but together with, of course, the touristic season, make this seasonal trends. And these are the data, the concentration addressed in the constitution. We have almost a proof, okay, that the evaluation of the contaminant burden in muscles may be quite effective. Also in the short time, because here we are talking about 1 year, you know, and one pre-season, may be quite effective in looking at the trends also of contamination and over regional scale. Looking at the biological data set, I told you that we selected laboratory experiment, but with a very wide array of experimental setups, so different species, different chemicals, different experimental setup, different life stages of the species. So there is really a huge amount of results that are also very, very diverse. Okay, so we should, before starting doing what we would like to do with the following weeks, I hope, months, maybe. We should try to understand how this biological set is composed. And we did the concurrence network analysis by using the keywords that are included in the title and also as keywords in the papers. So the 148 studies gives at the end 221 useful keywords. So we eliminate all the keywords that were too general. So just pharmaceutical type, therapeutic class, biological endpoint, and invertebrate species. And we also consider the analysis to the keywords that co-occurred for a minimum of 3 times. And at the end, this is our biological data set with the main keywords that are reported and the linkage between them. So from this, which seems like mess, no? Say, okay, so what we have in the end, but we could like navigate inside this mess and try to see, for example, the main biological models employed that are, of course, the muscles, the medium cells and tapes, so the cramps, pollicator, like a discrete versicolor, that is a very popular model to study the sedimentary pattern of the crustacean for the effect related to behavior like movement and stop. And then the main therapeutic classes investigated, like non-steroidal anti-inflammatory drug, ibuprofen, paracetamol, the psychiatric drugs like the carbamazepine and fluoxetine, and caffeine, the stimulant. And there you see also the connection between the other keywords. and where they are. And more interesting for us, the main physiological processes investigated. Metabolism stand out as the main, absolutely the main investigated. Oxidative stress, lipid peroxidation, that is always involved in the oxidative stress. Neurotoxicity, the psychiatric drugs have been the subject of analysis for a lot of studies on neurotoxicity, but also for the function, okay, of the nervous systems of invertebrates, in particular encephalitis, that are not included here, but also in the benthic invertebrates. And the production that included also the studies on embryos and so the embryo development. So now, from this analysis, we should focus on these terms, bivalves, the anti-inflammatory drugs, and as another point, the metabolism and the oxidative stress. Actually, there are a lot of papers that also address the function, so the therapeutic effect of the pharmaceuticals that will be included in the shortcoming meta-analysis implementation, that is a way, that you see here, to integrate and analyze the results from multiple studies and really see which are the pharmaceuticals that are more effective on bivalve, which are the species that probably show more sensitivity or more tolerance to this kind of pollution, and also to see where are the complication barriers. So on a regional scale or on a therapeutic or species scales. And so where to implement the result. Indeed, just for this, that is actually preliminary analysis, we can see that there are some discordances between the field and the lab studies. In the field, we have different species that show pharmaceutical accumulation, and so that are potentially affected by the negative effect of pollutants. In the lab, we have just a few of these species that have been actually analyzed for this topic, and so maybe more study on ecologically relevant species to be performed. And by, bypassing the fact that we all use the same animals, because we are sentinels, we know very well the physiological workings and so on. So bivalves are excellent models study, but they are not the only one, and shouldn't be the only one. And then again, also for the therapeutic agents analyzed, there are really a lot of data in the field that say that antibiotics, psychiatric drugs, anti-inflammatory drugs are widespread in the environment, in the animals, but also in other environmental matrices. But the biological studies just focus on anti-inflammatory drugs, psychiatric, something and antibiotic, and for example, antibiotics that are really a worldwide problem are not really addressed today biologically. They are quite massively, I have to say, studied, for example, for the problem of the spread of antibiotic resistance in SD, but also in animals, but not for their effect on the animal. Okay? And the data that we have said that they actually have. And we have for sure some bias in communication, because the environment is almost studied by other areas really less represented. Maybe also for political problem, and also for all the southern Mediterranean, which is lack area for the what concerning environmental issues, so we don't know which are the point sources of fire and how this kind of environmental issue are, and probably they are not answered because they have other kind of problem in this moment, unfortunately. And then also the biological process investigated, because we have a lot of data on the effect of metabolism, oxidative stress, because they are the conventional biomarkers that are always explored biochemical assay, and also the reproduction. But there are still few data for the so-called mode of action approach, and also few data on those physiological processes that may be quite relevant and key for the wellness, not for the status of some specific animals. And I consider, for example, the shell classification of bivalves and other hard tissue forming invertebrates. And this is quite important for this kind of new approach that has been employed to analyze the effect of pharmaceuticals as the adverse outcome. So the adverse outcome pathway, or RP, that is easier, is an experimental approach that wanted to put as an order, okay, in the array of effects that we see in the organism, given by the interaction with the body. And of course, it included also the mode of action, because the initiating event of the possible outcome for the pharmaceutical effect on the organism is the mode of action of the pharmaceutical. So the interaction with the specific cellular target to which the pharmaceutical is related. And then we can have a series of key events that stands behind the molecular initiating event and the final physiological outcome. So this event may be at the cellular, at the tissue, or at the organ level, up to the adverse outcome that can be that are always huge physiological impairment, like impairment of reproduction, impaired development, death of the animal. Okay, this is the very, very final outcome. But the impairment of reproduction, for example, may then have effect at the population level. So it's the therapeutic effect of this of the molecular initiating event. So some AOP have been already developed, like for fluoxetine, that I told you is the active principle of product. That is an antidepressant, very famous. The action of serotonin is to block the reuptake of serotonin in at the presynaptic level here. And so by increasing the concentration of serotonin, the can bind its specific receptor and start the cellular event that mediates mode of action. So it's a sort of indirect way to normalize okay, with certain margins in patients that have a certain level of simulated test. So this is the molecular insensitive event. As to the marine vertebrate, we don't really know if this serotonin transporter are indeed present in the neuronal cells. But for sure we know that in different tissues we have some serotonin receptor expressed, and that this receptor works in a similar way as the human receptor, similar, not the same, but similar. So it is possible, okay, it's absolutely likely that the velocity may accept on invertebrates the same chemical function that is bacterial. And indeed, we were able to build, so to monitor the series of killers that we detect of the oxygen, of course, of serotonin. This was also a good, I think I have the graph, yes, yeah. It was a good way also to understand much more about the circulatory system. So it was a good output on the basic results with animals. So now, for example, we know that the receptor that mediate the effect of serotonin, oxytocin, in the acid down masters is a type 1 serotonin receptor, which has a series of mediator inside the cells. And the final outcome measured not only on levels, for example, also on substance, so the adverse outcome is reduction of metabolism, alteration of red cell growth, reproduction, alteration of the reproduction system. So spawning, the spawning between males and females of different species. In fact, problem. Reduction of the reproductive output in terms of this, and also alteration in the development operation, behavior and locomotion affecting substance, the operation of the locomotion behavior, and of course, alteration of the substance. So, since the level of oxygen in the coastal areas, in some coastal areas are quite remarkable, this may be an environmental problem. The other environmental problem is given by the panel, which is a compounds that is used to treat cardiovascular disease, and it is really highly described with the active principle of Ingera, which is probably the anti-target drugs mostly described worldwide. There are a lot of data concerning the influence partner, also because it's one of those chemicals that is stabilized at the pH and salinity levels of water. So it changed this chemical feature going from the water, and it has been maybe the first pharmaceutical that showed how the motor function approach is, how much important is the motor function. Because if you see the output of the conventional physiological test that are employed to address the environmental risk of chemical compound, we can see that the concentration of compound that are effective no, are quite higher than the levels that are measured in aquatic environment, both seawater. So we could conclude that propanol is not an environmental threat. But if you see the kind of biological endpoint that we analyzed, you can see that are all apical endpoints. So endpoints at the individual level. And the concentration of compounds that induce a significant effect on this endpoint are usually quite higher compared to the molecular endpoints that make the whole factor. Indeed, by addressing, so we went in vivo studies down in the concentration, so with the studying lower concentration of the higher. Covered all the spectra of the environmental burden, but we went also lower and beyond. And we saw that, first of all, by the propanol, they act as conventional, so we can use a stress. This is one of those biomarkers that have been included in the Italian and also the European legislation for the environmental monitoring. It is a lysosomal stability. That is a very popular biomarker of exposure to stress because it comes with the general stuff of the organism, and it is really easy to perform. You can, I did it yesterday on my lab practices with students, and how to do this. Because it is done on leukocyte cells of the muscles, so we just take the element of the cylinder and we expose the cell to colorant, that is an invertebrate, that can penetrate inside the cells and inside the lysosome. And if the membrane of the lysosome is intact, cannot flow outside. If the membrane of the lysosome has some degradation, it can leak out from the lysosome and disperse the cells. So we basically measure the velocity, so the time to which the cells lose the red coloration. So this is why you see our time. This is the literal representation time, which is the time at which the 50% of the cells that you have in the slide have lost the red coloration because of the stabilization of the lysosomal membrane. And the more this time is smaller, no, the more the membrane were destabilized. And so the air start of the animal to accident. So the controls are generally very high stabilization times, that means that the air coloration was lost very slowly, while the highest concentration, you see that they have a compression time significantly reduced compared to the drugs, which means that the final set this configuration quite important stress effects. Talking about the mode of action, no? Propanol as a therapeutic agent is a beta adrenergic receptor. It means that it blocks the receptors that mediate the effect of the catecholamines like adrenaline and adrenaline. And it is linked to a series of cell mediator of its effect. I'm trying to keep it as simple as can, so that I don't want to detail with the chemistry of facility. So this beta adrenergic receptor may have different functions for yes, this beta adrenergic receptor may mediate different function noradrenaline according to the history they are expressed. So we have regulation of metabolism, reproduction, GLP functioning, the stress response, and the regulation of the genes. Concomitantly and unfortunately, propanol is also an antagonist for the serotonin receptor that we just saw before. So according to the proportion of adrenergic or serotoninergic control of the tissue of the organs that we are addressing in our masters or other invertebrates, we may have a different outcomes of the problem. That it is always related to the its mode function. For example, here we saw the completely different effect of the exposure of the water in the panel, but in the digestive gland, where we have the prevalent of the energy control and the mantle, the muscles, that's the biggest part, in which we have equivalent supplement. This is because the beta-adrenergic receptors are positively coupled with this intermediate molecules of the signaling, while the serotoninergic receptors are negatively coupled. So by blocking the adrenergic receptor, we push the negative effect of the signaling, while by blocking the serotonin receptor in microdonuts, we eliminate the negative regulation that constrain the signaling. The molecules that we are addressing that are the levels of the cyclic activity, the cells that include structural reduction and the activity of the protein scale are the same, the protocol are the same, the difference is execution. And also the animals are the same. So these mantras and the digestive one have the same levels. So this is just really a tissue effect related to the population of membrane receptors that are exactly related on the tissue development. The fact that this compound interacts with neurotransmitters like serotonin or adrenaline, it is also a problem when it was a problem when we started scientific experiment, because these results of the first two are probably the second or the third attempt with the in the first stage, we did this experiment. Okay, so we started with just an integration, a very smaller, in the experiment, but unfortunately, we did the experiment in October, November, when the animals reacted to the reduction. So they have a completely different neurodevelopment aspect compared to the February March when we repeat this experiment. At this stage, we did not obtain any kind of effect. That was pretty strange because, of course, we could predict, at least the digestive gland output. What is my pointer? Yeah, at least the output of the digestive gland from, of course, what we know, what we knew about the. So it was quite strange that the cycling concentration was not changed and was not reduced in the digestive gland with exposure to the environment. We did the same experiment on lymphocytes, not in October, but within these 2 days, and we got what the results that we expected. So we decided to repeat the experiment under different different reproductive stages. And in this case, in both cases actually, we measured the lysosomal membrane stability, that is a test that we always do when we take the muscle from the seed, just to see how they are, okay, if they are fine, or if there is some problem. We got last summer, we put the muscle, we did the unilateral function test, and we observed that it was not really found. I called some colleagues at the Alpha, and they told me that we had a toxin, alpha toxin involved. So it was not really good. It was for an experiment for an animal that we already discussed. So this is a good way of performing the experiment in not suitable condition. In this case, of course, the lysosomal membrane stability was not so terrible, but it was not as good as here. Here we also look at the condition of the guards because we have the suspect not going correctly here, and that it was related with the production, and actually it was low. So by repeating the experiment, in different reproductive condition of the animals, we got the result that showed. So this is quite important. So that the correct period of the year with this kind of experiment with the essential evidence to eliminate the patterns. And of course, now we do the experiment always, not in the same period of the year, but by looking at some parameters that are related with that stuff. Because we know that with this changing climates, we cannot really be sure that it will February, March. Okay. Are you okay? Relax. Okay, we will finish early. Okay, so we can go actually. As I told you, some pharmaceuticals may have effect on physiological processes that are essential for some organs, like the biomineralization. Chemicals like serotonin or dopamine, they that address basically the neuro endocrine control of the muscle physiology, may have effect on basically all the physiological processes and farming. Now, studying the changes in visualization potential in the adults, it's quite challenging because it requires very long experiments because of the slow growth rate, but the latter stages in this are very helpful to overcome this experimental issue because, as I told you, they go from the fertilized egg to the first classified shells in 24 hours. So they are very popular models to study the effect of environmental on the mechanism, okay, very molecular mechanism that control biomineralization and cell biology in humans. Okay, these results are from some years ago, but they are really clear, no? To see which are the effects of a very popular anti-inflammatory drug, that is the clofenac, which is the active principle of the anti-inflammatories and the series, okay, of the pharmaceuticals. These are the same microscope pictures of the shells of our veg that have grown with 1 microgram per liter of diclofenac, which is a 3-fold concentration. And you see how this the shells are florid or present breakage and other kind of. And also we did some analysis on the content of calcite and analyzed and so that the shell have been almost totally demineralized. So the diclofenac likely interact with the embryos by disrupting the molecular machinery that lead to the formation of the pioneers. Yes, this is the carbonic RNA amylase and protein synthase, which are unregulated by one of the. And the same is Smodalion, which is the most popular in the factor. And also in this case, we have like the signal by the last test. The most proportion of the malformed cells were embryos that were not able to become better, so there is a delay in development, the down of the development, but this was expected by considering this is a disaster. The less expected stuff were the strong down regulation of genes involved in biomineralization of again, and we also have that we are strongly down. But remember, it's not, I think it's not a large scale, it's the normal scale stuff. The expression will be a lot of the that's relation. Oh, I've done. Yeah, I ran a little bit. Just to notice, since I know that you are always looking for a congress or workshop, the CTAC, which is the Society for Environmental Toxicology and Chemistry, which is a very huge organization. Just to imagine that the congress is divided into the continent, so we have the CTAC in the US, the Europe, the Asia. It's a very, very big congress. So the Congress of CTAC in Europe this year will be in Bologna, in Italy. Yes, but every year, more or less in October, we organize a congress, a small workshop of the Italian language branch of CTAC, which this year will be in Naples, on October 13 and 15, 2 days. That is open, of course, to all people that work on issues like related to competition. So not only chemical contaminants, but for example, there's kind of changes. And it is a very open to young users. So there is also a contest for the young person. The winner will win the inscription to the CTAC Europe of 2026. The people subscription to this congress are quite high, so it's a very good answer. for the student also to have the possibility to attend this kind of so interdisciplinary and very interdisciplinary conferences, there is everything exactly. So people that work on change, people that work on life cycle assessment, and also people involved in the economic aspects of environmental protection, environmental assessment, and so on. So if you want to attend the workshop and then the congress, it's good. We also have one the teacher that attended 2 weeks ago, so if you want to ask her, she classified further. So in the podium, but not the first, unfortunately. But I think it was a good experience for us. For her, also to present the data of the PhD project, people that is in the field, also from the university. So I think all of you if you possibility. So if you are interested, you can contact me, and also I think there is a Facebook page that on the Facebook with all the details. Now we are now finalizing the session. There will be a session on kind of changes, but also or quite likely a session on the conservation of biodiversity. Okay, so I'm done. Do you have questions? Since you've been in silence for the talk. Also in Italian, if you are kind of English, it's not a problem. One. Yeah.
Do you feel that the aspect of the document is a lot of environment could also happen to completely other environmental organisms, or it's something related to the knowledge to the environment?
No, I think that okay, since I think that the effect on environmental regulation are likely the result of the fact that this compound interacted with the regulatory pathway that are really highly concerned with regulation. And this is why we are able to perform that kind of effect on masses. Because by the way, we should be able to and we don't have to. But and also that the target, the molecular target is so conserved that there is the huge possibility I'm sure that was wrong. No, I'm quite sure because I think that the already is so. And this is why it is important, for example, to perform mode of action studies of organisms different from the conventional mode that we employed as part of the masses and more ecologically relevant, no, than physiologically relevant. Then we could add the problem of studying the regulatory pathway with the receptor to the around. But maybe it was also the story of the assets. Because when we started that, we didn't know what is not. So we started recording the experiment with the adults and see, okay, we saw an effect. Oh, segment is reduced, but also in the humans, it always happens that if the survival cells and express the better storyline with the segment is reduced. So probably the regulatory pathway is the same. Okay, so we go to study the regulatory pathway. And so we started with the in vitro experiment that I didn't show here, but of course it's completely long story. on the analysis of the regulatory pathway. And we saw that so it was a lack of trust because the process vigorous results, but it's not so good the animals, of course. The regulatory pathway is concerned. It works not as a human, but almost. And I cannot think that this is just a particular feature like the mass of the organisms. I'm quite sure that maybe there will be some differences, of course, but only the lost scale of the regulatory pathway concerned. Other questions? Yeah, absolutely. And there's back to another black today this afternoon and okay, so if you have questions, so you can do another question after. We think we can finish here. Thank you. I do not I don't see hands raised.
Okay.
No, there are some people. I said people that looks connected up there. Okay, so if you don't have questions, thank you for your attention. And we can finish here. Thank you.
Thank you.
Thank you.